Hello everyone, topic of today is cell injury. So it is the part 1 of cell injury from Robbins. So सबसे पहला topic है what are the causes of cell injury. So the causes of cell injury can be divided into various divisions. First is hypoxia, then you have agents which are physical and chemical agents, then you have infectious agent 2, then you have immunological reactions and genetic abnormalities. So you can remember these enumerates by the word Hagi. From H we have hypoxia, A we have agents, G we have genetic abnormalities, I we have immunological reactions. In hypoxia we further have 3 causes, which is ischemia, cardiorespiratory failure, decreased oxygen carrying capacity. And the causes of this decreased oxygen carrying capacity are further divided into 3: anemia, carbon monoxide poisoning, and increased blood loss. In physical agents, we have increase or decrease in temperature, increase or decrease in atmospheric pressure. In chemical agents, we have increase in salt or glucose concentration, increase in oxygen which leads to oxygen toxicity, arsenic poisoning, cyanide poisoning, or mercury poisoning. In infectious agents, we have bacterial or fungal infection. In genetic abnormalities, there can be 2 things. Either the enzyme formation can decrease, which leads to decreased protein formation, or if there is much more protein, it can lead to accumulation of damaged DNA or misfolded protein. After that, we have types of cell injury. So there are reversible and irreversible. Before diving into the detail of each, firstly we will see the general overview. Reversible injury, so as the name suggests, it can be reversed. So what are the basic things which we see in reversible injury? We can understand this by this simple diagram. So first of all, what happens in the reversible cell injury? There is clumped chromatin. Second, there is generalized swelling of the cell and the organelle. So this was a normal cell, now there is a swelling. Third, there is blebbing of plasma membrane. Fourth, there is detachment of ribosomes from the endoplasmic reticulum. So as we saw, there was generalized swelling, there was blebbing of the plasma membrane, clumped chromatin, detachment of the ribosomes. In liver we see fatty changes, which is accumulation of triglyceride filled vacuoles in liver, and this swelling leads to pallor, increased turgor pressure, and increased organ weight. When this swelling occurs in endoplasmic reticulum, there is pinching of endoplasmic reticulum, which leads to formation of vacuoles. This is known as the hydropic change or the vacuolar degeneration. In the reversible cell injury, we see red cell because there is eosinophilia. Apart from these, there are various microscopical changes. First is plasma membrane alteration. As we saw, there was blebbing. Next, there is mitochondrial changes. So what is this? When the cell membrane is damaged, the phospholipids form the myelin figures in the cytosol. So in cytosol there is accumulation of myelin figures. Then there is dilation of endoplasmic reticulum, and there is also nuclear alterations. So now this is the first image. This is the normal kidney tubule, as we can see. In this image there is the swelling of the cells, and there is eosinophilia in the cytoplasm. So this indicates reversible cell injury. So after having an overview of reversible cell injury, we will move on to discuss some basic things about necrosis, apoptosis, and necroptosis. All of these 3 are the forms of cell death. So necrosis is accidental, means it was not pre-planned or it was not regulated. In this, the cell components fall apart. So because of this, there is local inflammatory response, and that local inflammatory response clears everything, and this is non-regulated. Apoptosis is the opposite. It is regulated and there is no inflammatory response or any collateral damage. Another one is necroptosis. So it is kind of mixture of these two. The detail we will study afterwards. So in necroptosis, it is a pathway which is genetically controlled which genetically controls some form of necrosis. Okay, so now let's see necrosis. So necrosis is the pathological process which is a consequence of severe injury. So the cause can be ischemia, any microbial toxin, any chemical or physical injury, or when protease leaked out. So when this protease leak out, it can occur in pancreatitis. So these are the causes of necrosis. Necrosis is accidental cell injury, consequence of severe injury. So what happens in necrosis? There is denaturation of the cellular proteins, and then there is leakage of the cellular contents which lead to inflammation. So we see local inflammation, and after that there is enzymatic digestion of the injured cell. So from where does this enzyme comes? When the plasma membrane is damaged, the lysosomal enzymes come in cytoplasm, which leads to the digestion of the injured cell. So now, injured cells have DAMPs on it. DAMPs is damage associated molecular products. So now these are of ATP from the mitochondria is also a DAMP, and uric acid from the DNA is also a DAMP. So they attach to the receptor in the macrophage. So after attaching to the receptor in the macrophage, two things happen. First is phagocytosis, second is release of cytokines, and the inflammatory cells release proteolytic enzyme. So now this phagocytosis and the enzymatic digestion leads to clearance. Okay, so in necrosis there is plasma membrane damage which leads to protein leakage, and these proteins can be detected in blood tests, so they are known as biomarkers. Okay, so why is it irreversible? Because the mitochondrial dysfunction can't be reversed and there is profound disturbance in the membrane function. What is the morphology of necrosis? There is increased eosinophilia because of release of proteolytic enzymes. Then it is glassy because of the glycogen loss. Then we see vacuolated cytoplasm because the organelles are digested, and we also see myelin figures as we discussed earlier. So now, in the myelin figures, dead cells are replaced by the phospholipid precipitate. So now what happens to these myelin figures or the phospholipid precipitate? They are either phagocytosed or they are converted to fatty acid, which then undergo calcification. In necrosis, we also see discontinuous membranes, mitochondrial dilation with increased amorphous densities, and aggregation of fluffy material, which is the denatured protein. So now we come to the nuclear changes in the necrosis. There are three. First is the karyolysis. In karyolysis, the endonuclease decrease the DNA, which leads to basophilia being faded. So in karyolysis, the DNA decrease because of the endonuclease activity, and the chromatin's basophilia is faded. Second is pycnosis. Nucleus shrinks, which leads to chromatin condensation to the dense basophilic mass. So here the basophilic mass increases. Then we see karyorexis. In this, there is fragmentation of the pycnotic nucleus. So now we come to type of necrosis. The first one is the coagulative necrosis. In the coagulative necrosis, the architecture of the dead tissue is preserved for some days, and the affected tissue has a firm texture. So what happens in it? Because of the injury, the structural proteins are denatured and the enzymes are also denatured. So the proteolysis of the dead cell is blocked, and because of this we see intensely eosinophilic cells with some reddish nuclei which may persist for days or weeks. So what happens after these? The necrotic cells are broken down by the action of lysosomal enzyme, which are derived from the leukocyte, and these also remove the debris of the dead cells by phagocytosis, the mechanism we discussed earlier. So what are the examples of coagulative necrosis? Ischemia caused by obstruction in the wall in all the organs except brain. So except brain, ischemia of every organ leads to coagulative necrosis, and a localized area of coagulative necrosis is known as infarct. So now let's see the image. So this is the gross image where we see the wedge-shaped infarct in the kidney. So this is a coagulative necrosis. This is a histological image of kidney. So we can say that this is the glomerulus whose outlines are preserved, and we can see severe eosinophilia. So these are the things we see in coagulative necrosis. The next thing we have is liquefactive necrosis. In this, what happens, there is digestion of the dead cell, which leads to transformation of tissue into a viscous liquid. So where do we see it? We see it in the focal bacterial or the fungal infection, because the microbes stimulate accumulation of leukocytes and the liberation of enzyme of the cells. So what happens, that here leukocyte comes after a few days and then the cell is digested. But here bacteria or the fungal, they stimulate the accumulation of leukocyte, and it leads to digestion of the dead cell. So the necrotic material seems creamy yellow because there is presence of leukocyte, and this material is known as pus. And where is it seen? It is seen in brain, in the CNS. This is the image of liquefactive necrosis in the brain. So we can see that there is dissolution of this brain tissue. Then we have the gangrenous necrosis. It is not a specific pattern of cell death, but it is a very commonly used term. So generally it is used for the lower limb. So when it is used, when it is lost at blood supply and it undergoes necrosis. So typically it is a type of coagulative necrosis. So when the bacterial infection is superimposed, there is more of a liquefactive necrosis because the bacteria attracts the degradative enzymes. So what is this? So whenever there is such type of infection, it is known as wet gangrene. So this gangrenous necrosis can be both coagulative or liquefactive necrosis. So it is not something different. Other we have the caseous necrosis. So caseous necrosis, the most common example is tuberculosis infection. Caseous means cheese-like. So cheese-like means the white appearance of the area of the necrosis. So whenever we do the microscopic examination, what do we see? We see structureless collection of fragmented cells which have some amorphous granular debris which is enclosed in an inflammatory border, and this appearance is known as granuloma. So let's see it. This is the caseous necrosis. We can see a cheese-like white network. So this is an example of tuberculosis. Then we have fat necrosis. So what is fat necrosis? Here we see fat destruction. So how are the fats destroyed? Because the pancreatic lipases are activated and they are sent to pancreas and the peritoneal cavity. So in which condition does this happen? In acute pancreatitis. So here the pancreatic enzyme leaked out from the damaged cell and they liquefy the membrane of the fat cell in the peritoneum, which will release the triglycerides, which are now split by pancreatic lipase. And now these fatty acids combine with calcium to produce chalky white areas. So on histological examination, we see shadowy outlines of the necrotic fat cells along with some basophilic calcium deposits and an inflammatory reaction. So this is how fat necrosis look like. We have the chalky appearance. Another thing which we have is fibrinoid necrosis. So we see this type of damage in the immune reaction involving the blood vessels. So it occurs when complex of antigen and antibody are deposited in the walls of arteries. So when they are deposited along with the plasma protein which has leaked out of the vessel, we see a bright pink and amorphous appearance which we call a fibrinoid appearance. So this is the fibrinoid necrosis. So we can see a bright pink area of necrosis with inflammation. Okay, so the last topic of day is that the necrotic cells would disappear by enzymatic digestion and phagocytosis. If they are not destroyed, there is deposition of calcium which leads to dystrophic calcification. So that was all for today. Thank you for watching it. So bye.